Now let's talk a little bit more about that glycosidic bond that holds together the nitrogenous base onto the sugar itself. I briefly showed it to you, and I talked a little bit about the forms of it being a beta form, and I showed you where it attaches. This is a N-glycosidic bond. So we saw in sugars attach to each other also through a glycosidic bond, but they have an O-glycosidic bond, meaning when you have one sugar, for example, attached to another sugar, it forms this bond through an oxygen, right? That is an O-glycosidic bond. For our nucleotides, our sugar uses its anomeric carbon right here, but it forms a bond to the nitrogen on our nitrogenous base. So whatever that base may be, this nitrogen highlighted in either the purine or pyrimidine is the part that is actually attaching to the anomeric carbon on the sugar base. As I also said, this bond is configured in a way to make it a beta configuration. And remember, that means that on the sugar itself, the bond on the anomeric carbon, this N-glycosidic bond, is on the same side as that CH2OH group. And this group actually is what attaches to our phosphate. So these are on the same side, meaning they're either both pointed up or both pointed down. They're not on opposite sides. That is what the beta means, remember, when we are talking about this bond. Now again, this nitrogen attaches to this bond, this N-glycosidic bond attaches to a particular point on the pyrimidine versus the purines, and that's either the N1 position on a pyrimidine or the N9 position on a purine. So to highlight that on a purine versus pyrimidine, you can see here on our pyrimidine, this is the N1 position on our pyrimidine, and that is where we attach the bond for our base sugar, our pentose base sugar. And for something that has a purine, like this purine here, you can see that the nitrogen here on the 5-membered ring portion is what attaches through this N-glycosidic bond down to our anomeric carbon of the base sugar. Now, this whole group that is attached to the sugar actually has free rotation. It's almost like a flag that is waving around on the sugar base. This normally is rotated in a manner that can either be syn or anti. You can see the syn is where they are stacked over each other, where the anti is where they are. This is kind of flagged out to the opposite. This is the same also for a group in which you have the pyrimidine type group top. Now let's talk about variations of the nitrogenous bases in our nucleotides. One type of variation is a tautomeric structure of the nitrogenous base that already exists. This is a special type of tautomer, which is a prototypic tautomer. This is just a structural isomer, structural meaning movements of actual bonds, so bond places have changed, that differs only based on the location of a proton. So we're just moving our protons around. One very common form of this type of tautomerism is the ketoenol. We've talked about this most likely in organic chemistry, where you have, for example, some ketone. Here's our ketone. It's attached to other stuff. And this bond here moves. So our double bond to our oxygen moves to form a double bond between the two carbons. And in doing that, this hydrogen here moves onto our oxygen. So we have movement of the hydrogen in a ketoenol tautomerism. This is another type of prototypic tautomer, which is called the Lactam tautomerism. And it occurs in some heterocycles, including in our nitrogenous bases. Now, both of these versions that I'm going to show you do exist in solution, but the Lactam form is the predominant form at a neutral pH. When we're talking about biological pH, the Lactam form is predominant. Just like when we talk about in normal pH environments, the keto form of a ketoenol tautomerism is usually the predominant form. Here's what that lactam tautomerism looks like. Here is uracil, and you can see its lactam form. This is where we have two carbonyls here versus the lactim form. It's very similar to the ketoenol. You can see our double bond has shifted from the carbonyl on the top to inside the ring. And this hydrogen has shifted to our oxygen. There's also a double lactim form in which you have another shift of a hydrogen. This is on the other carbonyl. So you can see after our first shift, this carbonyl and this hydrogen can then also shift in our double lactim form, forming a double bond here instead of on the carbonyl, and our hydrogen moving onto the carbonyl oxygen. Why is the lactam form the more predominant, or why hopefully it is at a pH of 7? Well, remember that this nitrogen, the N1 nitrogen on our pyrimidines, is the one that is utilized in binding to the actual sugar through that N-glycosidic bond. So something like a double lactam form is very unlikely, whereas this lactam is slightly more likely, where you have just our carbonyl at the top shifting its hydrogen, but still the lactam form, the form that we have you memorize, is the form that we are likely going to be seeing at a pH of neutral. But it's important to know that this is happening because that makes for interesting chemistry that can occur at this portion of our nucleic acids and nucleotides.